Hi, I'm Dr. Zoe Waller, and I'm an Associate Professor in Drug Discovery at UCL School of Pharmacy. This video is to explain our recent research paper, hopefully, in a simple way. It's just been published in a journal called Nature Communications. The links to the paper are included on the YouTube webpage. The title of the paper is Structural Insights into i-Motif DNA Structures in Sequences from the Insulin-linked Polymorphic Region. The work is very much a team effort from multiple experts and my colleagues within UCL School of Pharmacy. So let's start at the beginning to get some context. You might already know that our bodies are made up of different types of cells, and in each cell, our genetic information is held in the form of DNA. DNA is made up of 4 building blocks, which we call bases, A, T, C, and G, otherwise known as adenine, thymine, cytosine, and guanine. These 4 building blocks encode the information to make life on Earth. The code to make a human being, for example, is 3 billion letters long. In DNA, 2 strands of the building blocks coil together to form what is known as a double helix. This structure is a bit like a twisted ladder, where the links between the strands, or the rungs of the ladder, hold them together. However, depending on the sequence of the building blocks, DNA can change into different structures. DNA, which is composed mainly of the base G or guanine, can form unusual structures such as G-quadruplexes. And DNA, which is composed mainly of the base C or cytosine, can form unusual structures such as i-motifs. These both look very different to the iconic double helix. Our team are particularly interested in i-motifs, which are the ones that form in sequences that contain a lot of the base cytosine. Alternative DNA structures are thought to potentially play a role in the development of genetic diseases such as cancer and diabetes. We are particularly interested in the potential changing shape of DNA in diabetes. Diabetes is a disease that occurs either when the pancreas does not produce enough insulin or when the body cannot effectively use the insulin that it produces. Insulin itself is a hormone that helps glucose in your blood enter cells in your muscle, fat, and liver, where it's used for energy. This is part of the DNA sequence for the insulin gene. The letters in bold show the genetic code for making the hormone insulin. In advance of this code for making insulin, there is a region of DNA which is known as a variable number of tandem repeats. For this gene, it's called the insulin-linked polymorphic region. It was previously known that this region of DNA in the insulin gene can fold into alternative DNA structures and shapes. It was also known that this region of DNA varies between people in both sequence and length. This means you and I may have different insulin-linked polymorphic regions. This type of repeating region is actually quite common in the genome, but this one in the insulin gene helps control the making of insulin. Let's look a little bit closer at some of the sequences which vary between people. These sequences initially may look the same. But if we colour the differences in the sequence, it's easier to see them. As you can see, the differences between these different sequences are actually very small. Our work shows that these different variants in sequence fold into different DNA shapes. For example, these sequences fold into hairpin-like structures. And these sequences fold into i-motifs. In our model experiments, we found different shaped DNAs affect whether insulin is switched on or off. When we looked at the sequences that folded into G-quadruplexes and i-motifs, we saw insulin being expressed or switched on. Whereas when the sequences folded into hairpin structures, there was no expression of insulin. We wanted to learn more about these different structures, and we were lucky enough to be able to crystallize the full-stranded DNA structure called an i-motif. Our crystals allowed us to determine exactly what the structure of this DNA looks like. This has revealed that certain DNA sequences have special additional interactions which help them form alternative DNA structures like the i-motif more easily. Now we know more about the structure. This research means that we can now use the shape of DNA to design molecules to bind these structures and affect insulin expression. These could be developed into drugs and potentially medicines. As this is the first crystal structure of this type, it will also be useful as a model for other targets in the genome which form this shape of DNA. This research was funded by Diabetes UK. If you have any questions, please feel free to ask either by tweeting me or by email. Thank you very much for watching, and bye-bye for now.